I want to give you today an update about something that is much less fancy than clinical trials and gene therapy in 28 products. I just want to speak with you about what we need to be ready to get the drugs, not only on the market, but for every single patient everywhere in the world. So I've got a couple of disclosures. So we sometimes believe that we conduct a clinical trial, and the clinical trial is successful, then the patients have access to drug. Easy. It's a little bit more complex. The clinical trial, if it's successful, needs to go to be presented as a product to the regulators. We'll look at the methodology of the trial, the outcome measures, many things, by the way. And if the drug is approved in several countries, you need to go to a second animal, which is called the payer. And the payer will look at different outcomes, like the value for money, the disease burden. And that's only after this round that the patients could get access to the drug. And it's not guaranteed that all patients will have access to the drug, because it may happen that in some countries, a drug is approved, but not reimbursed. That's the case of several drugs in Europe, right? To name some, for instance, in Belgium, Ateluren is approved but not reimbursed, which means that technically no child is on Ateluren, right? And because what the payers are looking in a filing is not the same that what the regulators are looking at. And what is important also to understand is that according to how we have designed our plan, the payers in several countries could give access to drug to a certain number of patients, to a certain category of patients, especially if the trials has been conducted in a very well-defined and restricted category of patients. So how can we ensure that this is successful? Well, believe me, I have a long track record of failures. So I know what I'm talking about in terms of failures. In Duchenne muscular dystrophy, we have been conducting trials for 15 years now. And we don't have a drug that has clearly demonstrated its efficacy in a double-blind randomized placebo-controlled study. We have drugs that are approved, but for which we have no formal proof of efficacy. Spinal muscular atrophy was a complete different story. And we have learned how things may happen when we are successful, with all the beauty and all the ugly side of the success. So if we want to be ready for discussing with the regulators everywhere in the world, to discuss with the payers everywhere in the world, we need to understand our outcome measures. We need to understand the methodology of the trials, the value for money, the disease burden, which means we need to have natural history studies. And please note the big S. It's not a single one we need several. Because we need first to be sure and to confirm what we find in a natural history study. And we need to collect data all over the world. And that's how we can validate outcome measures. And outcome measures, by the way, can be also, for the sensitivity to change, be validated during clinical trials. But we need to appreciate that the collection of data and the good collection of data during prospective, well-controlled studies are super important. For those of you who could not attend on Thursday during ABOM, it was so clear the importance of natural history studies to design the clinical development plan, but also to validate the outcome measures and to understand the variability over 1 year, especially when there is a placebo effect. So to give you a little bit of update, indeed, I've been recruited by force, by Alison, to meet fantastic people of FAST UK. And we initiated our natural history study in Oxford. And it was not even started that several countries asked us our protocol. And yeah, we could give it in Belgium, in Hungary, in France, and in Spain. And we know, for instance, that in Belgium, we will recruit our first patient soon, thanks to a grant of Fondation Léon Frédéric. In Spain also they are ready, and Hungary is also moving. So yeah, that's Europe. But one step further, little step further, we could send our protocol to our good friends in LATAM, and in South America. They are incredibly active and committed people to make things moving there in South America. And there is a very active community that needs to be supported. So we could send our protocol to Fast Latam, to Argentina also, and to Australia. And entered in discussion with two pharma companies also to share the data and to share the protocol, so that our Oxford NHS protocol has been shared so far with 7 academic patients advocacy and 2 pharma companies, which demonstrate the value of the investment of FAST UK. It's not only in one country that you prepare the future. We don't need a natural history study. We need several natural history studies to demonstrate that we have a robust set of data. So now, what are the first learnings of the patients we included? I'm not going to give you the baseline of the few patients we have included so far, but I can tell you what is not in the database, what is in our day-to-day basis. The first thing is that those natural history studies are burdensome, and clinical trials, I can tell you, are burdensome. And even if we try to limit as much as possible the number of scales and assessment, then if we want to assess the patients on every single aspect, including speech and behavior and ambulation, we need to perform a lot of tests, which is very burdensome for the children. And it takes time if you want to do it properly. The worst of the assessment by far, in my opinion, is the EEG. We have used wearable outcomes. We have made ECG during clinical trials. We have made several blood tests, but the worst was definitely EEG. That's probably in the most burdensome exam during clinical trials in natural history study. We learned that and when I say, I mean, all of this is new for me, so sorry to state the obvious for you who are in the field for so many years. But, you know, for instance, when you conduct trials in Duchenne or SMA, you put EMLA everywhere, you know, cream and then patches everywhere on the skin. We discovered, we found very quickly that the patients, Angelman patients, most of the time they are much more disturbed by getting the cream and on the skin everywhere during 1 hour, rather than getting the punction at first. And then we thought that it was a good deal to organize it on 2 days, and it is actually a good deal to organize all this assessment on 2 days. But to sleep in an hotel when you have a patient with Angelman, but you know it better than me, can be very disturbing for the patient. And many parents prefer to sleep at home even if there are troubles. So that's the very few things we learned, and which are so different of the neuromuscular world. Then about the wearable device, as you know, one of the reasons why we had so many failures in Duchenne is certainly the poor outcome measures that we have been using, and that we are still using, by the way. Wearable devices have the opportunity to provide a much more holistic assessment of the patients, and to appreciate the patients over a much longer period of time passively. We thought initially that wearable devices were not very suitable for patients with Angelman, and they would never accept that. It's true if they wear it on the wrist, but on the ankle, it's absolutely not a problem. So the Wearable device, to give you on which I'm working, has been updated for the hard drive. Don't call it Actimio anymore, call it Side. It is now much lighter, much slower, much smaller. The volume has decreased. I think it's super important for our patients with multiple sclerosis or Parkinson or spinal muscular atrophy. But to be completely honest, I'm not sure it's going to change a lot for patients with Angelman, because when it's on the ankle, it's not such an issue. Maybe it could help for trying to get some assessment in the wrist, but I'm not sure that it's going to increase the incidence for the ankle. Then the outcomes. We are working on several outcomes, starting for those that we know quite well, which is the stride velocity. And we know that patients with Angelman, first of all, they have a slower stride velocity, but also with a regularity of strides that is very different of the one that you observe in the controls, with much less variability. We're studying walking perimeter, and we believe that it's an important outcome to figure out what you can actually do during real life, and what you can engage in terms of social activity. Then force, or kneeling, which is the way our kids move on the floor to pick up some toys who could appear to be full, which are not full. And then we try to measure ataxia by the time spent on one foot rather than 2 feet when you are walking, and the walking deviation of the foot. Some of these outcomes now are very well understood, and we have all what we need in terms of reliability of time of recording. Some are a little bit less characterized, and we're still acquiring data. But the good news is that we are acquiring quite a number of data in 2 academic natural history studies that are ongoing, one that is initiated, one pharma NHS that is planning, and 3 therapeutic trials that are either recruiting, either in planning. So that makes so far an increasing number of hours recorded in Angelman syndrome. So we have today 9,000 hours of recording in these patients, and this makes a gold mine in terms of data to try to figure out what is the best outcome, what is the most reliable, what is the outcome that gives us the best chances to be successful in the clinical development. And of course, we need to move on the regulatory perspective. There is a letter of intent that we are preparing for the FDA, and that we hope will be sent early next year. So I just want to finish this update by thanking the people. That was the day of my capture. Look, those people from Fast UK and Alison, my team in Oxford, my team in Liège, and which is, you cannot see it on the photo, but we are all wearing our Angel mask. So in 2020, be 20, travel by train, use your bike, and more importantly, wear your Angel mask. I really look forward this year. I think that we are going to get much more data, much more results, and the way the things are moving in this field is just incredible. I'm super happy and proud to be with you today, and unfortunately remotely, and look forward to the year that is coming. Thank you very much for your attention.